Yes. So first you should draw the box around the secondary clarifier. You see that 5,000 meters per cube is leaving from one side. And then from the downside, half of whatever is coming to the wastewater treatment plant is leaving, and half of whatever is coming is 2,500, right? From this side, 5,000 is leaving. From this side, 2,500 is leaving. So which means what is coming? 7,500, in fact, right? Yes. So that.
So I don't. Okay.
So now, if you recalculate, you should get a better answer.
Let me recalculate.
Yeah, go ahead.
I got 34 now.
34?
Mm-hmm.
Not exactly what we want, but maybe correct. Just give me some time. I had the value here. Yes. So also your V0 is 0.75, but look at the units. It's meters per hour, right? But your flow rate is meters cube per day. You see?
Oh, 24.
Yes. You should multiply by 24, and then you should get like 18, I guess. Yes, you will get 18. And then if you divide 7,500 by 18, you will get the area. And from the area, you can calculate the radius. And from the radius, you can get the diameter. Sevinç, Homoyen, how is it going for you guys? Are you lost? Do you need my help?
I am getting there slowly, but...
Okay. Area 0.42 meters. No. It's very slow. It's very low for the area. It's not even 1 meter squared. It cannot be like this. So tell me, Sevinç, what is your flow rate? Q. What Q you used? Yes. And what is your V0? But V0 that you calculated in the beginning. I know that the formula is Q over A, but the number of V0 that you got in the beginning. Okay, so look. So the first thing that you should have started from is finding V0. So you see that here I sent in the, so is my screen in fact visible? Yes, my screen is visible, so you should be able to follow me. So here you see, in order to find the area, you need to have V0. And this V0 you can get from SVLR. SVLR, you already know, it was given in the question. It is 0.3. X, you know, it is 4. SVI, you know, it's given in the question, 100. And from this, you will find V0. And your V0 will be some number. And you should find this first. Go ahead. The Q is 7,500. Why? Because you have the clarifier, this secondary clarifier, and you should just make a balance. So you see that from one side, the wastewater that is leaving the clarifier is 5,000 from one side. From the bottom, 2,500 is leaving. So if 5,000 leaving from one side, 2,500 leaving from the bottom, then 7,500 should be coming in, right? So 7,500 is coming in, 2,500 living, 5,000 living. So your Q, in fact, will be 7,500. Lafos Humayun, can you also turn on your cameras so that at least I could see you? Thank you, Sevinç.
Yes, yes.
So the first thing that you do is you find V0 by this formula. 0.75, not 750. So first action. Then the second action will be to convert V0 from meters cubed from hours to days, because your flow rate is in days. So V0 times 24 in order to convert, which will be 0.75 times 24. It will be something around 18. So the load was meters cubed per meters squared per hour, not per day. So now this is the second action. Now that you know V0, you can find area by just dividing the flow rate by the area. But first you should find the flow rate. Third action, find flow rate. And flow rate will be 2,500 plus 5,000, which will be 7,500 meters cubed per day. V0. So your SVLR is 0.37 inch. You multiply it by 1,000, and then you divide it by X, which is 4, and you divide it by 100, which is SVI 100, and you get 0.75, right? And once you find the flow rate, your force action, 0.1, what, V0?
I guess we should like divide by 0.1 because in units it's written like milligrams, yes?
It's my fault. I guess it was my typo. Sorry, Sevinç. If you look at the screenshot of the problem, it's milligrams per gram.
Like without M, like liter per grams, yes?
No, with in the question screenshot, it's milligrams per gram. But if you're confused about this M, milliliter, then this M will be taken by this 1,000 in the SVI test. By this 1,000, this empirical number that is calculated using this SVLR, this 1,000 will take care of this milli thing. And in the units, everything will be correct then. Yes, and our force action will be to find the area, which will be simply Q divided by V0, Q divided by V0.
7,500 divided by 18, right?
Yes. It should be 416.66666. So just 67, we'll say for now. And the fifth action will be to find the radius from this area, which will be 11.51 meters. Yes, here. So it will be just 11.51, and the diameter will be somewhere, yeah, 23, 24, but we don't usually have 23 diameters, so I guess in practice you would just make it 25. Yes. So here it is. Okay, we go on with the lecture. So now the radius is 11.5, or the area is 416.67. And then we have our height. Let's choose it to be 2 meters. And so now we know the area, we know the height, so we can find the volume, which will be simply 416.67 times 2 meters. It will be 833 meters cubed, right? So the total volume of our secondary clarifier will be 833.34. Great, we found it. We did it. Yes, now. Let's come back. The calculation has ended. What I will do is, if right now it's still a little bit like vague for you, I have a very good video that actually explains how it's done. So I'll send that. If now it was a bit complex, right? Okay. So bulking sludge. So do you remember this SVI thing? This SVI test? So usually if something wrong happens with our bacteria, what it does not settle. So after 30 minutes, you will not get this clear picture, but you will still end up with this thing. It will not settle. And it's very, very bad when our bacteria does not settle. Why? Because then we cannot clarify. We cannot separate water from our bacteria no more. We cannot do it anymore. And you will end up with this picture. And at the end, your clarifier will look like this. Do you remember that these are, in fact, the bars of the secondary clarifier that are staying in these edges? And in fact, not this, but clear water should be falling from here, right? But when our bacteria does not settle by gravity, but swims, maybe, right, inside of the water, all over the water, this will be what we are getting. And at the end, if it doesn't settle, it will just wash out with the water and it will not be separated, which means we basically did not really clear our water from any BOD or COD. And my hypothesis that I promised you to tell. So one day when I was going back from Salar Wastewater Treatment Plant, so I'm no longer doing internship there, but I go there once in a while in order to get some information or ask for something. And last time I saw this picture here. So you can open this, and when you open this, so basically this, what you're seeing here is one of the tubes. So Salar Wastewater Treatment Plant, once it cleans the water, it sends it to the Salar River. You see this Salar River, and this tube that is opening, in fact, is discharging the water, the treated water, to the river. And in fact, the treated water should be similar in color, in odor, in chemical and biological concentration to the river's water. But you can clearly see, you can notice that the color is different. And if you watch the videos that I sent yesterday, that you may notice that in fact the color looks very similar to the color of the water that was in the biological treatment place, you see? So what is coming from the treatment place, and in fact you can see a clear difference of the color of the river water and the color of the water that is coming from the plant. And I assume, so I took this photo in order to ask the lab people there about what might have happened, why this water is looking like this. I forgot to send, and this picture was just in my gallery until today. And I thought like maybe this will be good to show this to you. Yes, so you can see what might happen. So my assumption is that they were faced with this bulking sludge problem when their bacteria would not settle. So what it means? In fact, if you will look at this bacteria from the microscope, then at the end, you want your bacteria to be like this. So they are grouped together, they're bulky, they're dense, and they are in flux, so they can settle easily. So here you can see that they can settle easily. This one is in groups, in bulks. However, at times, if the influent to the wastewater treatment plant changes somehow, what happens is that wrong bacteria starts to grow. And this wrong bacteria looks like this. So you see that they have a lot of hairy stuff growing from them. So it's different filaments. And in fact, what this filament does, it doesn't let the bacteria group in flocks. And what it does, it makes bacteria very porous. It will be porous, it will be light, and it will not settle down. And at the end, we will end up with this kind of picture. So when does it usually happen? So there are a lot of reasons that may cause this problem. And there are a lot of bacteria that may grow from this. And usually this might happen if your oxygen supply to a secondary clarifier is low, meaning low dissolved oxygen, or you have very high nutrient availability, meaning nitrogen and phosphorus, there are a lot of them, or long chain fatty acids, meaning, for example, when such kind of places as Evas or Orte par lavage, they fry a lot of freeze, right? And they have a lot of oil and they just discharge it. And if someone took chemistry classes, you know that this oil is in fact long fatty acids, right? And what happened is that because of this, then you will face such kind of growth. And or at times, if your wastewater has a lot of sulfate inside, this kind of bacteria might grow. Or if your wastewater will have low pH, or there will be a nutrient deficiency, you might have some fungi that is growing, right? So usually when it happens, it doesn't happen always. It might happen if there is emergency somewhere and they had to send their wastewater to you, and that wastewater was not good. Or someone is breaking law and is sending the wastewater that should not be sent to you, right? So in such kind of cases, you will end up with this massive problems, and it's very bad for your treatment plant. And usually when such kind of thing happens, the immediate thing that you can do first is add aluminum sulfate. So if you watched the video of Ahmad, he was showing flocculation. He couldn't do it with aluminum. He did it with iron, but usually in practice we use aluminum. It's a very good flocculant, or you can use other coagulants. So what they do is they will force the bacteria to form these clots, and they will finally settle to the bottom. Or the harsh method, if you don't have this, the harsh method would be just add hydrogen chloride or peroxide to final clarifier. It will kill some of the bacteria inside, and once their volume will decrease, you will hope that they will go to the down, or they will at least populate the bottom half of the clarifier, not upper half. Bottom half, yes, not upper half. Or the extreme might be you will have to remove your sludge, and you will have to change it with the new one. Someone gave a question. Oh, okay. Yes. And however, you may be proactive. You may be proactive, and you can solve this problem while you're making this plant. And how different people solve it? So they usually add selectors. So what happens in selectors is, if you saw here, in fact, if your oxygen supply is very low, or your BOD, so this F to M, food to microorganism ratio, meaning BOD is low, then such kind of bacteria start growing. And what they do is, before clarifier, they will have these 3 selectors, and in 3 selectors they will make the opposite of those conditions. Their oxygen supply will be high and the BOD will be high, and when BOD and oxygen supply is high, you will not have those filaments, and you will avoid this problem. So you will be ready for whatever shock load that will come to you, right? So you will be ready for this. Or another way that you can do is so here you're giving oxygen, but you can not give oxygen if you want. You can make it anaerobic. In anaerobic, you will not have those, so whatever bacteria filament that could grow will stop growing. They will die. If they die, they cannot reproduce anymore, right? If they will not make spores, of course. So yes, these are the ways that you can prevent this problem right in the design part. Yes, and now the biological treatment part finishes. The only thing that you would like to know again is aeration, meaning how much oxygen you should give, whether you should give a lot, small amount, what kind of equipment should you choose for the oxygen supply. So all of those things. But I think we would not be able to fit this into this lecture. That's why I removed. Maybe I'll give it again with phosphorus and nitrogen removal so that you could watch later. However, today we will be talking about the chemical treatment and the chemical treatment that is going on in Salar wastewater treatment plant. So basically, there are a lot of ways to chemically treat the wastewater. And the chemical treatment, the purpose of chemical treatment is to get rid of whatever is left. And usually what is left is pathogens, meaning E. coli. E. coli will not die primary treatment. E. coli will not die in biological treatment, right? Even if some other bacteria will eat it, right? It can reproduce. It can get more and more and more. And what kills E. coli, it is a chemical treatment. And usually many countries, apart from oxidation with sodium hypochlorite, what you can use is ozonation. So you will just send a lot of ozone, or you will just send oxygen and you will give a lot of electrical shock to your water, and oxygen with electricity, it will produce ozone, and the ozone will just oxidize whatever is left in your water. Or you can use UV light. UV light with radiation will kill a lot of bacteria too. In fact, when we were covering Singapore uses a lot of UV light in order to treat the water. The ozonation is not recommended. Any chemical oxidation is not recommended. Why? Because when you oxidize, you don't know what kind of end product you might have, right? So for example, you're oxidizing E. coli into other molecules. The best oxidation happens when E. coli turns into CO2 and water. The most harmless products, CO2 and water. Water stays, CO2 goes. However, you should be warned against when the oxidation goes not till the end, meaning you might end up with other products. And what if those products are even more toxic than E. coli? And the worst part, you don't even know what kind of products you have. That's why usually in wastewater treatment, we are warned against using any oxidation processes. Yeah, it's better to capture and then kill rather than oxidize. However, we do so in Salar wastewater treatment plant, and usually what they do is they order a lot of technical salt. So this is basically just sodium chloride, and the sodium chloride is technical, meaning it's not cleaned. It will be dirty. It will look brownish. So, and they will take this salt and they will pour it into electrolyzing cells. In electrolyzing cells, this kind of processes, in theory, will happen. And at the end, from sodium chloride with electricity and water, you will have sodium hypochlorite. And sodium hypochlorite is very, very strong oxidating agent. It's in fact used in bleach as well, but with calcium, not with sodium, I guess, and it will oxidize a lot of bacteria. And in fact, you saw that Salar is really, really using it in a good way because their efficiency was like way bigger, right? 99.99938, right? So they use this method. Now I have some videos that I can share with you. These are the videos that I took in Salar, and in many of them, like this, in first two, you should be able to see how they add hypochlorite to the water. So now in this videos, you can see that they use it it's very primitive. Design is very primitive. I don't really know how it's done in other countries and whether they use hypochlorite at all. But here in Tashkent, they just have these tubes where hypochlorite is run, and they have a control overflow, so they can send a lot of, or they can send a small amount, and based on the E. coli concentration, they change it. So yeah, and in that way, and in this way, they do this process. And in the last video, you can see how the treated water is leaving the wastewater treatment plant, and it will travel further to river Salar. Yes. Yes, so by this our lecture finishes. You are welcome to take a look at Ahmad Khan's video, which is very, very interesting. Otherwise, our lecture finishes. And today we finished biological treatment, we finished chemical treatment, primary treatment, and in fact, the whole process of wastewater treatment was covered. Hopefully. The only part that is left is how we deal with the human waste, with this poop. How we do it, how we treat it, how is this utilized, what can be done with it. And it will be most interesting for the Lafrouz, Guliora, and Alpomish, because their research is going to concentrate on that. Yes, and hopefully we will cover this tomorrow, and I really hope that it will be very, very interesting, because I hope that I will have time to look at my old Soviet book that I sent you somewhere in the chat, because there people really wanted to make a food out of it, you know. So interesting. Let's see, and hopefully I will add some of that content to our book, and hopefully you guys will be able to use it in your research too. So thank you for your presence. Thank you for you being active. And I hope we nailed this lecture down. Sorry for a lot of calculations, and bye.